Meiosis is a process in which the number of chromosomes per cell split in half through the separation of homologous chromosomes in a diploid cell. The process of meiosis produces haploid cells that develop into gametes for asexual reproduction.
The male gametee is often referred to as a sperm, and the female gametee is an egg. A gametee is just a haploid sex cell.
A haploid cell is a single set of chromosomes, whereas a diploid cell is a cell with two sets of homologous chromosomes.
In a haploid cell, we only see one chromosome from each parent present, and in a diploid cell, there's two chromosomes from each parent.
You can tell how many chromosomes are present based on the number of centromeres. Centromeres is the middle of a chromosome in which chromatins sometimes attach.
This is a picture of two sister chromatids. A sister chromatid is when a chromosome duplicates and attaches to the replication.
During meiosis, a homologous pair gets cut in half. A homologous pair is the two sets of corresponding chromosomes found in a diploid cell.
In a homologous pair, there is a tetrad, which is 4 chromatids, 2 from the mom and 2 from the dad. A chromosome is genetic information bundled into packages of DNA.
In this example of meiosis, yellow represents the mom chromosome and the blue represents the dad chromosome.
Meiosis starts in interphase and the cell spends most of its time there.
During early interphase, the cell does its cell functions and obtains nutrients to then grow. There is no arrangement and chromatins are arranged at random.
Chromosomes are replicated and paired with corresponding homologous chromosomes in the late interphase to transfer DNA.
The cell begins to divide and the chromosomes pair up in prophase 1. They also begin to cross over. Crossing over is when chromosomes pair together and pieces of the homologous chromosomes are exchanged.
Crossing over ensures that no person is alike because new combinations occur each time.
Metaphase 1 pairs homologous chromosomes and lines them up across the center of the cell.
Anaphase 1 pulls each homologous chromosome pairs towards the opposite end of each cell.
During telophase 1, the chromosomes then form a nuclear membrane around each cluster.
During cytokinesis, the two groups split and form their own two new cells.
In prophase 2, their chromosomes, each having 2 chromatins, become visible.
They line up in the middle of the cell and they are still diploid at this point.
In anaphase 2, the paired chromatins separate and this is now a haploid cell.
During telophase 2, the chromosomes then form a nuclear membrane around each group.
The clusters then split and become 4 new haploid cells, and this is the end of meiosis 2. Mendel's law of segregation and meiosis relate in the sense that in the law of segregation, each trait comes in pairs and is segregated into separate new reproductive cells. Homologous pairs of chromosomes are also separated in the process of meiosis.
Mendel's law of independent assortment relates to prophase 1 in meiosis. In independent assortment, some traits segregate independently from others, causing variation in probable outcomes. Also, in prophase 1, crossing over allows variations in genes that are in the gametes.